So, how in the world do you fight an enemy that you can't even see? Well, when an animal gets mysteriously sick, that's the exact challenge vets are up against. They have to become microbe detectives, hunting for a bacterial culprit that's totally invisible to the naked eye. This is how they crack the case.
Okay, so here's our game plan for this whole investigation. First, we'll hit the crime scene and collect our evidence. Then, we're going to use a special staining technique to get our first big clue. After that, we'll actually grow the suspect to see how it behaves, run a few final tests to be absolutely sure of its identity, and then we'll talk about why all this detective work matters so much.
All right, first things first. Every single investigation starts by gathering evidence. And in the world of veterinary medicine, the crime scene is the sick animal itself. And the evidence? Well, it's a biological sample, a potential hideout for millions, and I mean millions, of microscopic suspects.
So what the vet collects really depends on the symptoms. You got a horse with a big swollen abscess from strangles, the vet's going to collect pus. A dairy cow with mastitis, a milk sample is the key piece of evidence there. And if the infection seems like it's spread everywhere, they'll take a blood sample. Each one of these is a critical piece of the puzzle that gets sent straight to the lab for a much closer look.
And now we're in the lab. This is where our microbe detectives pull out a seriously powerful forensic tool. You know, it's a technique that's been around since the 1880s, but it is still the single most important first step in identifying bacteria, the Gram stain.
The Gram stain is honestly pure genius in how simple it is. It's what we call a differential stain. That means it doesn't just color the bacteria so we can see them, it actually sorts them. Based on how their cell wall is built, it splits almost the entire bacterial world into two distinct teams, the Gram-positives and the Gram-negatives.
And this shows you exactly how it works. See, Gram-positive bacteria have this really thick, dense cell wall that basically traps the first dye, staining them a deep purple. But the Gram-negatives, they have a much thinner wall, so that purple dye just washes right out, and instead, they pick up the pink counterstain. So, purple or pink. Just like that, our massive list of potential suspects just got cut in half. So let's say our detectives see purple under the microscope. Now they know they're on the trail of a Gram-positive. They zoom in a little closer, and what do they see? Do the bacteria look like tiny little spheres all clumped together like a bunch of grapes? Bingo! That's the classic calling card of a staphylococcus.
But what if the whole slide is full of pink bacteria? Well, the investigation totally pivots. Now we're looking at a Gram-negative, and if those little pink shapes are rods, a prime suspect immediately jumps to mind. E. coli, a super common culprit behind all sorts of infections in animals.
Okay, knowing if a suspect is purple or pink is a huge lead. But, it's not enough to convict. So the next step is to isolate the bacteria from that original sample and actually grow them on a petri dish. This lets us watch how they behave, and that reveals their unique cultural fingerprints.
Now this is where it gets really interesting, because some bacteria just can't hide their destructive tendencies. This agar plate here actually contains blood. You see those clear halos around the colonies? That's something called beta hemolysis. The bacteria are literally tearing apart the red blood cells around them. This distinctive zone of destruction is a dead giveaway for suspects like certain species of Streptococcus.
Other suspects, though, leave an even more unique signature. This description from a lab manual, it's pointing to a seriously dangerous pathogen, Bacillus anthracus, the cause of anthrax. When you grow it on a plate, its colonies form this wild, branching pattern that looks eerily like the mythical Medusa's head. It is an unforgettable and, frankly, a terrifyingly beautiful clue.
So the evidence is really starting to pile up, right? We've got a Gram stain result, we have a distinctive culture profile, but for that definitive ID, the kind of certainty you need when an animal's life is on the line, the detectives need one final confirmation. And this is where biochemical tests come into play.
This right here is the CAMP test, and it's a beautiful example of how microbial teamwork can reveal an identity. You streak a known staphylococcus down the plate, and then you streak your unknown suspect at a right angle to it. If that suspect is Streptococcus galactea, a major cause of mastitis, it releases something that supercharges the staph toxin. And what do you get? This perfect arrowhead of destruction. It's a microbial bullseye. Case confirmed.
So after all of this meticulous work, the swabs, the stains, the cultures, why is identifying one single microscopic organism so incredibly important? What's the big deal?
Well, it's because getting the right identification is the key to getting the right response. It means the vet can choose the correct antibiotic, the one that the bacteria is actually vulnerable to. On a farm, it means stopping one single infection from exploding into a herd-wide outbreak, which protects both the animals and the farmer's livelihood.
And, maybe most importantly, this work protects us. A lot of these diseases are zoonotic, which means they can jump from animals to people. Identifying and controlling a pathogen like Bacillus anthracis in a cow is the first and most critical line of defense in preventing it from ever infecting humans. This isn't just about animal health anymore, it's about public health.
So from just a single drop of purple dye to a perfect arrowhead on a petri dish, this whole process just pulls back the curtain on the hidden world all around us. It's a powerful reminder that with the right scientific tools, it doesn't matter how small or how hidden a culprit may be, its secrets can always be brought to light.